From the time you collected the culture stool sample at your home, Candida continues to grow without any competition because 95% of the original microbes in the stool sample have died or have become dormant while being transported to the laboratory. Lacking any competition for food, Candida continues to proliferate during the incubation phase at the laboratory. This overgrowth during transportation and preparation fuels the common belief of many doctors and people alike of the deleterious health effects of Candida overgrowth in the body. This belief has resulted in the inappropriate use of Candida drugs, supplements, and diets. Hello, I'm Dr. David Peterson of the Functional Splachnology Institute. I'm going to read a white paper published by Dr. Richard Lord that changed the way I viewed Candida and how I treat those with Candida symptoms. This is Documented Limitations of Culture-Based Stool Assessment. In the last decade, DNA sequencing has played a pivotal role in our knowledge on microbial diversity, providing an outstanding tool for the detection, identification, and characterization of microorganisms. DNA assessment has greater efficiency, reliability, and reproducibility, as well as providing both qualitative and quantitative data. DNA analysis is the new gold standard for identification of bacteria in clinical microbiology and has greatly facilitated the identification and classification of intestinal microbiota composition. Much of this has been done with techniques based on 165R DNA gene, which includes fluorescent in situ hybridization, denaturing gradient gel electrophoresis, and temperature gradient gel electrophoresis. DNA analysis is the new gold standard for identification of bacteria in clinical microbiology and has greatly facilitated the identification and classification of the intestinal microbiota composition. So why does anyone use the culture method of stool analysis? Conventional bacteriological methods, such as microscopy and culture, have been used for analysis and/or quantification of the intestinal microbiota for decades. Barriers such as investment in new equipment and staff, as well as commitments to ongoing analysis and research collaborations, interfere with what many laboratories are able to offer. Translating science to acceptable standard clinical protocols take time. Culture is still readily available and reasonably accurate, so clinicians continue to use it even though there are known significant limitations, which include transport issues, indeterminate and presumptive quantification and identification, as well as subjectively demanding techniques. In this review, we discuss 6 major pitfalls of a conventional culture stool assessment. Number 1, transport issues. The primary issue with culture analysis is that of transportation from the patient's home to the laboratory. Since analysis is culture dependent, meaning they have to grow the bacteria, sample collection must be done using nutrient broth containers to keep as many bacteria or fungi, Candida, alive as possible during transportation. This broth allows continued growth of those species most satisfied with the broth medium during the transportation, until the sample is finally plated for the culture. This growth results in a significant change in the balance of microbes present, since some species will more actively grow at the expense of others. DNA analysis eliminates this problem by placing the species in formalin vials for transport. Formalin immediately kills all organisms, freezing the exact balance present at the time of collection. Since DNA identification is only looking for the genes of the microbiota, living species are not necessary. This technique allows the clinician to develop the most appropriate therapy based on the patient's true gut microbiota, resulting in better clinical results. Recent studies in our laboratory illustrate this situation. One stool sample was placed in 2 vials, one vial containing formalin and another a nutrient broth commonly used for transportation. Both vials were incubated for 3 days at room temperature. Then the DNA was extracted. The extract was incubated with 3 different restriction enzymes which cleave the DNA at the specific base pair sequences. See Figure 1. Figure 1. A restriction enzyme, or restriction endonuclease, is an enzyme that cuts double-strand DNA when recognizing a specific short nucleotide sequence. The restriction enzymes SAW3A endonuclease cleaves DNA strands when it finds the sequence 5-GATC and 3-CTAG. These fragments can then be amplified using PCR. The digestate was amplified by polymerase chain reaction, PCR, and then placed on an agarose gel plate, and the DNA fragments were electrophoretically separated. Since the restriction enzymes cleave the DNA only at specific base pair sequencing, the broths with the identical populations of microbes would produce the same patterns of banding in the electrophoretic runs. If, however, there were different amounts and types of microbiota in the 2 tubes, then the differences would appear in the banding patterns, and this was clearly demonstrated in the experiment. See Figure 2. Figure 2. 3 restrictive enzymes, HAE Roman numeral 3, MBOL, and SAU3A digest the same 2 samples. One sample comes from the culture and sensitivity vial containing a nutrient broth. The other sample contains formalin. Both vials have been incubated for 3 days. The banding patterns are significantly different for the 2 vials, indicating that populations of microbiota present are significantly different. In addition, the culture and sensitivity vials have much fewer and heavier bands, indicating that the microbial populations have reduced diversity due to anaerobic die-off, and those viable ones have overgrown. Some bands present in the formalin vials are not detected in the culture and sensitivity vials, where whole groups have died off. L is the calibration known as DNA sizes. Another observation from the data is the loss of banding in the nutrient broth culture and sensitivity vial over time, indicating an overgrowth of aerobes at the expense of the anaerobic populations and a consequent loss of diversity. Opportunistic, potentially pathogenic organisms can also overgrow under these conditions. This overgrowth has been seen in our laboratory. For these reasons, nutrient broth transport vials cannot provide the specimen integrity required for accurate measurement of gut microbes. Figures 3 through 5 illustrate the growth response of bifidobacteria, Candida species, and Staphylococcus aureus in one subject's sample over 3 days. This data indicates that the clinical laboratory data relying on the transport of the specimen to the laboratory for culture testing are likely erroneous and can lead to inappropriate patient treatment. Candida species overgrowth is particularly notable in Figure 5. As these data indicate, Candida proliferates in the nutrient broth transport medium. This overgrowth in transport has likely fueled the common belief of many practitioners of the deleterious effects of Candida albicans overgrowth in the gut. This belief has resulted in the inappropriate prescription of antifungal supplements and drugs. Figure 3. Transport effects on bifidobacteria concentrations. A significant decrease in bifidobacteria occurs over the 3-day incubation in the standard nutrient broth culture and sensitivity vials. Formalin fixed samples show more consistent levels. Figure 4. Transport effects on Staphylococcus aureus. Potentially pathogenic, the opportunistic bacteria Staphylococcus aureus shows significant overgrowth during the 3-day incubation in the nutrient broth culture and sensitivity vials. Formalin fixed samples show no growth. CFUs are colony forming units. Figure 5. Transport effects on Candida species. Candida species show significant overgrowth in nutrient broth culture and sensitivity vials over 3 days. Formalin fixed sample vials do not exhibit growth and are at a level that would not be reported as abnormal growth. The culture and sensitivity samples at 1, 2, and 3 days would be reported as abnormal overgrowth. This data indicates that clinical laboratory data relying on the transport of specimens to the laboratory for cultural analysis are likely erroneous and can lead to inappropriate patient treatments. 2. Quantitative assessment. In DNA assessments, quantification of predominant, opportunistic, and pathogenic bacteria are given as exact amounts. Yeast/fungal values are given as a +1, +2, +3, or +4 and represent a specific value range. For example, a +1 is specifically quantified to mean 100 to 999 picograms of DNA per gram with a systematic reproducible result. Similarly, bacterial assessments are quantitatively reported as colony forming units per gram of sample. In laboratories that use stool culture assessments, values of quantity are significantly less specific or reproducible. For example, when a stool sample comes into the laboratory, they are plated onto a dish with a selective or differential media. The technician dips a tool, generally a cotton-tipped applicator, into the stool sample and puts it onto a plate that is divided into 4 quadrants. The technician starts streaking in a quadrant 1, then drags the tool to quadrant 2, quadrant 3, and then quadrant 4 in sequence. The dishes are incubated overnight, at which time they are assessed. If the bacteria grew in the first quadrant only, it is called a +1. The assumption is there was only enough of the bacteria in the stool sample due to low concentrations to grow only in the first quadrant. The first quadrant is expected to have the greatest number of bacteria since that is where the streaking starts. If colonies appear in all 4 quadrants, it is called a +4. The fourth quadrant would be expected to have the least amount of the original bacteria present since it was the last streaked. Thus, if something grew in the fourth quadrant, it is assumed that it grew there because the bacteria were present in high concentrations in the sample. No procedure or assessment is done to confirm this assumption. No standardization for the original amount of the stool sample on the tool exists. A +4 theoretically means that there was enough of the bacteria on the stool sample that it stayed on the tool while it was dragged from the first to the fourth quadrant. However, with no standardization, these steps are at best a subjective judgment by the technician. Additionally, if growth is found in the first and fourth quadrant, but not in the second and third, some technicians may call this a +4, while others may call it a +2. These results are technician-based and allow for human error, such as the technician turning the tool differently in the last quadrant. Consequently, a +1, +2, +3, or +4 from culture samples is an unverified guess based on where the microbes grew in the culture dish. This is at best a qualitative assessment of bacterial presence in the sample. These discrepancies are some of the reasons hospital-based laboratories give results as a positive or negative and do not attempt to quantify when using culture techniques. 3. Presumptive diagnosis and identification. To identify individual microbial species growing on the initial plates, individual colonies are selected and grown out again. Using a process of elimination composed of several if-then steps, a series of tests are done which yield a diagnosis of the most likely organism present. Typical characteristics such as colony morphology, key biochemical reactions, and drug susceptibility patterns assist in establishing the supposititious identification of the microorganism. However, most labs imply that this diagnostic identification of the microbe is definitive. It is not definitive, but at best gives a percentage best estimate of the likelihood of identification accuracy. It is widely accepted that plate culturing techniques reveal only a small portion of the true microbial population, primarily due to an inability of detecting organisms that might not be culturable with known existing media, temperature specifics, phases of metabolic activity, and the inability of growing known microorganisms which are viable but enter a non-culturable state. DNA analysis became the standard of research more than a decade ago, particularly due to the subjective nature of culture identification systems. The sensitivity of conventional culture method was compared to DNA analysis for identifying Shigella species and enteroinvasive Escherichia coli in known cases of dysentery. DNA had an identification accuracy of 96% compared to the culture's identification rate of 54%. 4. Identification of predominant bacteria. Bifidobacteria are among the first to establish themselves in the infant's intestinal tract, and in the healthy infant, they predominate. While it would be impossible under normal life circumstances to have a +0 as a level of bifidobacteria, that level is often reported in laboratories utilizing culture techniques. In these laboratories, lactobacillus and bifidobacteria, both facultative anaerobes, are identified primarily based on selective growth media. Though further enzyme tests such as coagulase or catalase may be done to hone the identification of lactobacillus, bifidobacteria identification is done solely on this single growth. Quantification is also made from the growth on these plates as described in section 3. These phenotypic methods suffer from a lack of reproducibility generated by conditions of the stool culture used in different laboratories and to the diversity of strains that comprise the recognized species. DNA identification and quantification of lactobacillus and bifidobacteria is exact. 5. Parasitology. Parasitology is yet another field of microbiology to be greatly improved by PCR DNA testing. Classically, parasites have been identified by microscopy and enzyme immunoassays. In recent studies, DNA techniques have proven to be more sensitive and specific than classic laboratory methods. One example is Giardia. Since Giardia cysts are shed sporadically and the number may vary from day to day, laboratories have adopted multiple stool collections to help increase the identification rates for all parasite examinations. Even with the advent of antigen detection systems, there has long been uncertainty in the diagnosis when no ova or parasites were found during microscopic examination. With PCR technology, only one fecal sample is needed for 100% sensitivity and specificity in the parasite examinations. The clinician needs to be aware that assessment of parasites, while good by microscopy, is much more accurate with DNA-based technologies. 6. Automated identification systems. There are 3 main automated systems in use for microbial identification: the Microscan, Vitek 2, and Crystal GP. All use the if-then sequences discussed earlier to identify characteristics of microbiota and produce proposed bacterial identification with a given accuracy expressed as a percentage. The accuracy of these systems was evaluated by identifying coagulase-negative Staphylococci. Verification was done with DNA by 16S RNA sequencing. The Microscan, Vitek 2, and Crystal GP systems correctly identified 82.5%, 87.5%, and 67.5% of the isolates respectively. Misidentification was the main problem in the Microscan and Crystal GP systems, whereas the main problem of the Vitek 2 was low-level discrimination. None of these three phenotypic systems tested could accurately and reliably identify staphylococci at the species level as well as the 16S RNA technique. There are two types of Vitek systems, the Vitek 2 fluorescent card and the Vitek 2 colorimetric card assay. In another study, the accuracy of identification at the species level for the Vitek 2 fluorescent card, the Vitek 2 calorimetric card assays, and the API 20NE, another identification system, were 48%, 59%, and 46% respectively. In comparing the 16S RNA gene sequencing to the Vitek 2 fluorescent card, the Vitek 2 only identified 43% of the microbes correctly at the species level, a very low rate. Due to this low rate of identification, the authors proposed an algorithm for proper identification of non-fermenting gram-negative bacteria. In the algorithm, isolates with only good or acceptable identification to the species level should be subjected to 16R RNA gene sequencing when accurate species assignment is sought. DNA assessment is specific and accurate in the identification and quantification of the fecal microbiota. As discussed above, stool culture methods are relatively inefficient, time-consuming, and labor-intensive, often leaving the diagnosis obscure. DNA is highly sensitive and specific. Beginning with the stool sample collection, DNA assessment is superior because it avoids the pitfalls of transportation that the culture methods suffer from, and allows the identification of the true gut microbiota as well as the anaerobic population comprising 95% of the gut microbiota that go dormant or die in the presence of air. The results are the actual types and amounts at the time of collection, not what survived in the growth medium after incubation. DNA further reports the microbiota as true numbers, values that are specifically quantified, not an assumption. The ability of DNA to accurately identify microbiota has been one of the primary reasons why it has been so embraced by the research community for the last decade. Research on bifidobacteria and lactobacillus has significantly expanded since the introduction of the DNA analysis. DNA techniques are known to be more sensitive and specific than classic culture laboratory methods. Verification of the accuracy of the stool assessment tools and equipment is done by comparing their results to the actual value, which is determined by DNA assessment. Though culture stool testing has been correlated to clinical features in older research studies, we now know much of the data was incorrect. You win! You have Candida! Sellers of conventional culture stool tests argue that DNA assessment does not have the years of clinical background that culture has. This point, of course, is misleading. Though culture has been correlated to clinical features in older research studies, we now know much of the data was incorrect due to significant limitations of culture and the indeterminate results it produced. Researchers have been actively reevaluating past research for this reason. Specific values of gut bacteria have been identified for normal, healthy populations using DNA assessments, as well as for populations of selected groups and specific disease conditions. A notable example many clinicians are now aware of is that the research of Dr. Gordon and Dr. Lay, who discovered that obese volunteers had a greater amount of firmicutes and significantly less bacteroidetes than that of the lean participants. In their research on fat bugs, they performed a comparative 16S RNA gene sequence-based survey, or DNA analysis, of the distal colon gut microbiota, not culture. Gordon's team could not and would not have done this research with culture because it cannot produce this information. DNA analysis is the most thorough and reliable assessment of the gut microbiota. It eliminates the major limitations of conventional culture stool testing and offers advanced knowledge for more effective patient treatment.